over to my collaborator and colleague, Dr. Susan Jones. So before I start, I want to set the scene about our research. We're interested in fertility, and the broad definition of fertility is simply the ability to have offspring. And you can see from the graph of the world on the left here that the rates of fertility vary from country to country. And in Africa, the rates of fertility are higher than in other countries. So you can see on the right here that Zambia actually has an average fertility rate of 5 offspring per couple, but in some of the European countries, it's actually below 2. Now, the problem with fertility is that the rates are declining throughout the world. And here you can see different countries and different areas of the world in which the rate of fertility is declining. But you can see that a lot of areas now, the fertility rate is reaching below 2, which actually means that the population is going to be declining. And it's thought that if you extrapolate from this, by the time we get to the end of the century, most of the areas in the world will have a declining population rate. Now, you might think this is not a problem, but what it actually means if the rate is below 2 is that we're going to have a non-sustainable population. Now, the reasons for the decline in fertility are manifold. Some people are choosing to have fewer children. Some people are choosing to have no children. But there are some individuals and some couples that want to have children, but they can't have children. So their fertility is not through choice. So what is it about the control of fertility? Well, the control of fertility is from a part of the brain called the hypothalamus. And the hypothalamus, which is shown here, is located at the base of the brain, and it actually makes a connection with another part of the brain called the pituitary gland. So we're going to talk about the way in which the hypothalamus and the pituitary regulate the reproductive axes and control fertility. So this is referred to as the hypothalamic pituitary gonadal axes, and this is shown here in the male and the female. So if we think about the male first, which has the sort of most straightforward hypothalamic pituitary gonadal axes, the hypothalamus in the brain produces a GnRH, gonadotrophin releasing hormone, which acts on the anterior pituitary and stimulates that gland to make 2 hormones, luteinizing hormone and follicle stimulating hormone. These are called the gonadotrophic hormones, which are released into the bloodstream, and they can act on the testes to stimulate the production of sperm and also a sex steroid hormone called testosterone. Now, testosterone is released into the bloodstream, and it has this negative feedback action on the hypothalamus and the pituitary. So you have a standard negative feedback loop. In the female, it's a little bit more complicated, but you still have the central components, GnRH released from the hypothalamus acting on the anterior pituitary to produce LH and FSH, which obviously act on the ovaries of females to stimulate the production of eggs, and the sex steroid hormones estrogen and progesterone. Now, estrogen is quite interesting in that there's the standard negative feedback to suppress the axes, but at a certain point within the cycle, estrogen can become stimulatory. And this positive feedback action will stimulate the production of LH and bring about ovulation. So it's a little bit more complicated in females. Now, when we think about fertility, we have to think about when you become fertile. And the acquisition of fertility is at the process of during puberty, which is a period of emotional, behavioral, and anatomical development. After puberty, an individual becomes fertile. They can produce the gametes, that is, the sperm or the eggs. And the initiation of this process is happening in the hypothalamus, where you actually produce pulsatile gonadotrophin releasing hormone. And we're interested in what might be happening at puberty. What is the trigger happening in the hypothalamus to bring about puberty? So if you think about the GnRH neuron that's going to produce GnRH at puberty, before puberty, the activity of those neurons is suppressed, usually by GABAergic inhibition. So there's no GnRH output. But as we go through puberty, you remove this GABAergic inhibition and you acquire things that stimulate the GnRH neurons to produce GnRH output. One of these is glutamatergic stimulation, and another important factor that can stimulate GnRH output is this neuropeptide kisspeptin. So kisspeptin can act on these GnRH neurons to increase GnRH output, often in a pulsatile manner. There are populations of neurons within the hypothalamus that produce this kisspeptin, and these are called the KISS1 neurons. So at puberty, we get activation of GnRH release, brought about partly by the action of kisspeptin from the KISS1 neurons. A little bit more about what this neuropeptide kisspeptin is. So kisspeptin is a secreted protein, which is initially quite a large peptide, shortened by cleavage into a 54 amino acid, kisspeptin 54, and subsequently you can get even shorter forms of kisspeptin, either 14, 13, or 10 amino acids. And these are all as potent as the 54 amino acid. And any of these can stimulate GnRH neurons to produce GnRH. The neurons that make this kisspeptin neuropeptide are located in two different areas of the hypothalamus. So you find them in this anteroventral periventricular area of the hypothalamus, but only in females. Males have very little kisspeptin expression in this region of the hypothalamus. And you also find the kisspeptin neurons within the arcuate region of the hypothalamus of both males and females. So the kisspeptin neurons in the AVPV are sexually dimorphic. You only predominantly find them in the females, whereas in the arcuate, they're found in both males and females. And you'll see the significance of this shortly. So the kisspeptin neurons in these two regions interact with GnRH neurons and obviously stimulate the output of GnRH. The kisspeptin neurons in the AVPV region of females stimulate the cell bodies of GnRH neurons, whereas the kisspeptin neurons within the arcuate region act at the terminals of GnRH neurons. So they're acting at two different locations, these two different populations of kisspeptin neurons. The action of the kisspeptin neurons are that they signal through a specific G protein coupled receptor on the GnRH neurons. This receptor is called GPR54. So kisspeptin interacting with GPR54, you get subsequent coupling through a GQ protein, and you get a signal cascade within the GnRH neuron, which can cause a release of intracellular calcium, or through the action of diacylglycerol, you can modify the activity of specific ion channels within the GnRH neurons. Now, we've studied fertility and puberty using mutant mice as a model system, and we have generated mutant mice that lack the GPR54 receptor, or they lack the ability to make the kisspeptin neuropeptide. Now, both these mutant mice have exactly the same phenotype. That is, they fail to go through puberty. So here you can see a normal male mouse, and you can see that it's well developed. It's gone through sexual maturation. If you compare this with a mutant mouse, now this mouse is the same age as this one, but it has failed to go through puberty, and you can see that it has a micro penis and not very well developed testes. And both male and female mutant mice are sterile. If you look at the cause of the sterility and you examine the reproductive tract of these animals, you'll see that in the females, the mutant mice have got very thread-like uteri. This is the uterus in a wild type. It's very well developed, and the ovaries are very large. In a mutant female, you've got thin thread-like uteri, and the ovaries are much, much smaller. In males, in a wild type, you see a normal testes and a normal structure of the epididymis and the vas deferens, but in the mutant, the testes is very small and the epididymis is considerably reduced in size. And in fact, the mutants don't have the capacity to make sperm. So both the female and the male mutant animals are hypogonadic. They cannot release eggs and they cannot produce sperm. If you look at what the sex steroid levels are in these mutant mice, the males have considerably reduced testosterone levels, whereas the mutant females have got considerably reduced estrogen. So they have low sex steroids. And this is one of the reasons why they don't go through puberty. So if we now think about the way in which these KISS1 neurons integrate within the hypothalamic pituitary gonadal axes, this is what I've sort of shown you previously, but now we have the KISS1 neurons and how they communicate with the GnRH neurons. So on the right here, we have what we find in the male. So here you have the arcuate KISS peptin neurons, which are communicating with the GnRH neurons at their termini. And again, in the female, you have the same thing, the arcuate KISS peptin neurons communicating with the GnRH nerve terminals to cause an increase in release of GnRH. But only in the females do you have the AVPV KISS peptin neurons, which are communicating with the GnRH cell bodies. They're missing in the male. You don't need them in the male because these are the neurons that are involved in ovulation, and males do not need to ovulate. So now I'm going to pass over to my colleague, Susan Jones, who will present some of our unpublished data.
Thank you, Bill. So to summarize then what Bill has said, the neuronal processes involved in the initiation of puberty are driven by KISS peptin neurons. And this causes the pulsatile release of GnRH from the hypothalamus. And over the years, Bill and his lab have been instrumental in demonstrating the role of KISS peptin neurons in puberty and fertility. So when Bill and I began collaborating, we asked the question of whether these arcuate KISS peptin neurons, whilst going through this important developmental stage of puberty, do they change their properties? And so this brought to mind the idea of plasticity in neurons in the brain, which is seen in many brain systems during the development of neural systems. So by neuronal plasticity, we mean the persistent change in either brain structure or brain function. And this is known to occur both in the human brain and in the brains of the mouse and other types of animal models that we use in experimental science. So examples, very well-established examples of neuronal plasticity include changes in the activity of a neuron, which is referred to as action potential firing. And this involves changes in ion channels in the cell membrane, or changes in synaptic transmission between neurons. And this mainly affects neurotransmitter that's released and then acts on a downstream neuron. So in our talk today, we're going to focus on changes in the activity within a neuron, including action potential firing and ion channel changes. So the question that we're asking then is whether during the transition from infertility to fertility at puberty, is there neuronal plasticity in KISS peptin neurons? And the model that we're using is the mouse. So if we think about the lifetime of a mouse, when it's born, it goes through a period of development. And this is mostly complete around the time of weaning at about 3 weeks of age. The mice then enter what we might call a pre-puberty phase. Their hormones start to rise in preparation for entering puberty at about 4 weeks of age. The mice go through puberty between 4 to 6 weeks. And at the end of 6 weeks, we would say that the mice are post-puberty and they are now fertile. They can now reproduce. So once again, our question is, do the kisspeptin neurons, particularly in the arcuate nucleus, have immature properties when the mice are infertile and mature properties that are different when the mice are fertile? And so is there some kind of neuronal plasticity occurring during this transition phase? So in order to investigate intrinsic neuronal plasticity within arcuate kisspeptin neurons, our PhD student, Yuanjin Zhang, has carried out a series of experiments on kisspeptin neurons from mice, and we focused our work on females. So Yuanjin has recorded and obtained neurons from 2 populations of mice, those that are pre-puberty, so they're infertile, and those that are post-puberty or fertile. Yuanjin removes the brain of these mice and she prepares brain sections through the brain at the region where the hypothalamus is found and where the arcuate nucleus is found. And that's shown in this schematic here. So this is approximately where the kisspeptin neurons of the arcuate nucleus would be. So in this picture, we're showing you a real brain slice with the arcuate nucleus. And you can see here there are some red dots on in this region. And these are actually the kisspeptin neurons. They express a red fluorescent protein called TD tomato. So that enables us to see the neurons that are actually producing and releasing kisspeptin. If we zoom in on this region, we can see the fluorescence, this time in monochrome, so we're only seeing them in black and white. But these cells here that are lit up all contain TD tomato, and they're all kisspeptin neurons. So we can focus our study on this specific population of neurons. And Yuanjin has carried out 2 types of experiments during her PhD. The first is to isolate these kisspeptin neurons and examine their gene expression using quantitative PCR. And the second type of experiment is to do something called wholesale patch clamp recording, where she monitors the electrophysiological properties of these neurons. So neurons have a very important physiological property, which is that they can change their membrane status depending on whether they're resting or whether they're active and signaling. When they're resting, if you were to put a recording device called a patch pipette onto the cell membrane, you would be able to show that the membrane is primarily permeable to potassium ions. So potassium ions, when our cell membranes are resting, potassium ions are moving from inside the cell to the outside of the cell through potassium ion channels. But when our neurons are signaling, a different type of ion channel becomes important. This is the voltage-gated sodium channel, shown in yellow here inside the membrane. And under these conditions, sodium ions can move from outside of the cell to the inside of the cell. And when this happens, this generates an important voltage signal across the membrane that is called an action potential. And using our patch pipette on the cell membrane, we can detect these action potentials. And here's an example of a train of action potentials here recorded from a kisspeptin neuron. And so Yuanjin has recorded these action potentials, and she's counted the number of them and how frequent they are and how long the firing lasts for under different conditions. So if we look at that data then from 2 example kisspeptin neurons, this is a kisspeptin neuron taken from a mouse that was post-puberty and in the estrus phase of the estrus cycle. And you can see that there's a sustained train of action potential firing. If we look at a recording from another neuron. This time, this has been taken from a 3-week-old mouse. So this is before that mouse has gone through puberty. Again, it's a female mouse. So this is an infertile mouse. And now you can see that initially there is a train of action potentials, but these do not last for the full duration that we saw in the post-puberty mouse. So there's only a much shorter train of action potentials. So we've quantified 2 things here. We've quantified the number of action potentials that the neurons can fire and the duration of the firing. And if we have a look here, this is shown for pre-puberty mice at 2 different ages, but let's focus on the 3-week mice here and the post-puberty mice in purple here. And you can see that there's a significant increase in the number of action potentials, the maximum number of action potentials or spikes fired in neurons from the post-puberty mouse compared to those fired by the 3-week-old pre-puberty mouse. And this is accompanied by a much longer duration of firing. So this is the maximum duration of firing on the y-axis. And again, this is significantly higher in our post-puberty female mice compared to our 3-week-old pre-puberty mice. So we can conclude that arcuate nucleus kisspeptin neurons that are needed to activate puberty increase their maximum number of action potential spikes after puberty. Now, one obvious difference between a pre-puberty mouse and a post-puberty mouse is that hormones are present in post-puberty mice. So they've got circulating estrogen and progesterone, for example, and this will vary throughout the estrous cycle. I've shown you here a recording from a mouse in estrus when the estrogen levels would be expected to be quite high. So could this difference that we've shown you here simply be due to the amount of estrogen that's circulating? Yuanjin tested this by comparing the firing properties in estrus with a different stage of the estrus cycle in post-puberty mice. And that's the diestrus part of the cycle, where the estrogen levels would be expected to be lower. And actually, she found that there was not much difference in the firing during diestrus as compared to firing during estrus. And that's shown by this graph over here. The firing pattern is overlapping. So it suggests that the excitability of these kisspeptin neurons is not changing throughout the estrus cycle itself once the mice are past puberty and have become fertile. So to explore this a little bit further, we ask the question of whether estrogen is needed during puberty itself. So we've carried out 2 experiments to address this, and both of them involve removing the ovaries of the mice, which is the source of the estrogen. In the first experiment, we've removed the ovaries when the mice are 6 weeks of age, so just at the end of puberty, but just before they become fully fertile. So remember that the experiments I've shown you so far involve recording from normal mice that reach 3 weeks of age or normal mice that reach 6 to 8 weeks of age. In our experiment here, we have removed the ovaries at 6 weeks of age. We've given some estrogen to provide a flat, stable baseline of estrogen in post-puberty mice. And then we've recorded from these mice when they're 7 to 8 weeks old. So is there a difference in these mice that have had the ovariectomies, the ovaries removed at 6 weeks, compared to these 2 groups here? In fact, the animals that had the ovaries removed at 6 weeks are very similar still to the normal control post-puberty mice, and they do not show the immature firing properties that we saw in the 3-week-old mice. However, in a second set of experiments, we removed the ovaries at a different time point. So in this set of experiments, we're again comparing to our control mice at age 3 weeks and our control mice at age 6 to 8 weeks. But in our test group now, we've removed the ovaries at 3 weeks of age, and then we've left them to go through puberty with no estrogen present. And now when the mice reach 6 weeks, we give them the estrogen to flatten the estrogen levels in the post-puberty period. And then we record from them at 7 to 8 weeks. So what happens in these mice? Well, in this group, the firing properties more closely resemble the firing properties of the 3-week control animals and are not looking like the firing properties of the post-puberty control animals. So it suggests that removing the ovaries at 3 weeks of age and allowing the mice to go through puberty with no estrogen present is preventing this maturing of the firing properties of kisspeptin neurons. So just to summarize what I've shown you so far then, what we've shown is that arcuate kisspeptin neurons change their properties at puberty, change their activity properties at puberty. And this appears to require estrogen to be present at the puberty period between 3 and 6 weeks of age. So the next question we wanted to ask is, what is the mechanism of this form of plasticity that's occurring during puberty? Is it, for example, due to the ion channels in the membrane, such as the sodium and the potassium ion channels that I mentioned earlier, that are changing to bring about this? And in order to address this, we've used quantitative polymerase chain reaction to investigate changes in gene expression for the main ion channels that control cell excitability. And this work has been carried out by Leonie Paculat and Yuanjin Zhang in our lab. But we'd like to mention Dr. Stephen Manchiji, who did his PhD in Bill's lab and really set up the qPCR method in Bill's lab. I'm going to hand you back to Bill now, who will present these data.
Thank you, Sue, for presenting the electrophysiology data. So what Sue showed you is that there are electrophysiological changes in the kisspeptin neurons before and after puberty, and this seems to be mediated by a critical window of estrogen expression. What we wanted to do was to sort of try and understand what the molecular changes might be that are bringing about these electrophysiological changes. So we decided to use quantitative PCR to look at the expression of specific genes during this process. Now, before you can do this, you have to design primers that are specific for the genes of interest, and you have to validate these primers. And to do this, what you normally do is you isolate messenger RNA from the tissue of interest, you convert it into cDNA, and you use that cDNA to do your quantitative polymerase chain reaction, and you validate the various primers. So that's what we did initially. Once you have your primers validated and you know that they work and they amplify the gene of interest efficiently, you can then decide to look at the kisspeptin neurons themselves. So here we are purifying kisspeptin neurons. We're not looking at the whole of the hypothalamus. We're purifying kisspeptin neurons. We're taking the arcuate region from the hypothalamus. We're dissociating it using an enzyme called papain. And from those dissociated neurons, we are picking the specific kisspeptin neurons into small pools of about 12. So we're only looking in the kisspeptin neurons. And of course, we can identify them because they are fluorescent by that TD tomato. So you identify your pools of kisspeptin neurons, you make the RNA, and then you convert it to cDNA, and you do quantitative PCR for the target genes. Now, in order to do quantitation effectively, both looking before and after puberty, you have to somehow standardize between what's happening before and after puberty. You have to have a housekeeping gene which you can standardize against. So we spent quite a lot of time trying to identify a housekeeping gene that didn't show a change before and after puberty. There's no point standardizing to something that changes. So we looked at a lot of different housekeeping genes, and surprisingly, some of these housekeeping genes did show changes before and after puberty, so we couldn't use them. There were a couple that didn't seem to change. One of them was beta-2 microglobulin, and one of them is this transcription binding protein. So we decided to use the transcription binding protein as a housekeeping gene to quantify by qPCR. And we looked at the expression of some specific genes of interest. And here you can see some of the data we found for the genes that changed. We also looked at other genes which didn't change, but these are the interesting ones. And you can see that there's a voltage-gated sodium channel, which shows decreased expression after puberty, a voltage-gated potassium channel, a BK channel, which all show a decrease after puberty. And then there's an HCN1 channel, which actually goes up after puberty. So we think that these ion channel changes could contribute to the change in the neuronal excitability that we see. And we are following up on this, and we're actually looking at additional ion channels to see how they altered before and after puberty. So to summarize, what we have found is that during puberty, which is the process where fertility is achieved and you produce gametes, and this is driven by activation of kisspeptin neurons and an increase in GnRH. We now know that the kisspeptin neurons change their properties during this process. These changes seem to require an early estrogen between 3 to 6 weeks. And we've also found that there are specific ion channel changes that occur during this process. And we think that the changes that we find, the electrophysiological changes in firing, could represent maturation of the arcuate kisspep neurons being driven by these changes in specific ion channel expression. So I'd like to just thank the people that have been involved in this work, particularly Leonie Pucklet and Wanjin Zhang, who are 2 PhD students in my group and Sue's group, and also Stephen Manchisi, who was a PhD student and is now a lecturer at the University of Zambia, and we're continuing to collaborate with him to take this project forward. So thank you for your attention. Thank you very much.
Thank you, everyone, and we hope you have a really great rest of the conference, and we're sorry we can't be there.